Yes, and I won't talk about nuclear organization. I think it's a very nice. I think it's a kind of a trendy thing in epigenetics and epigenetic inheritance in particular, which I find very interesting. But I think it's the early days of chromatin organization in the field, I believe. So I first would like to thank you for inviting me to this day. I think there aren't many symposia completely dedicated to epigenetic inheritance, so it's always a pleasure to be part of such a rare event. And in particular events, symposia like this which cover different species, really from plants to mammals. I think it's really nice because it shows the diversity of the potential mechanism, which complicates the matter because, as many of you probably know, I mean, DNA methylation was one of the mechanisms which was always initially thought to be responsible, and now I think the view has evolved quite a lot, and one has to consider other mechanisms. So I think it gives really a very nice overview of the state of research in the field, which even for myself is extremely useful. So I'm going to tell you about epigenetic inheritance in the context of complex functions, so brain functions, behavior, because this is very relevant for the field of epigenetic inheritance, to try to understand the mechanism, but also because, I mean, mostly because it's highly relevant to the human being in relation to psychiatry, because there are many psychiatric diseases which are due to environmental exposure to the exposome, and which are transmitted across generations, but which mechanism of induction and perpetuation are not known. So disease risk is quite high for some diseases, and it's now there are many epidemiological studies in human which strongly suggest that disease risk can be transmitted independently of the DNA sequence. So there are really very large cohorts which, for historical reasons and other reasons, social reasons, have been put together and examined across generations. And some of these cohorts in relation to famine or in relation to nicotine or early stress, I mean Holocaust, induced or trauma linked to Holocaust. I mean, many of these studies have suggested that the conditions in which a human being, a child or an adult grows or live into for a certain while, which leaves traces on his or her body, can also induce symptoms, whether cardiovascular diseases for people who have been exposed to famine, or psychiatric disorders for people whose ancestors have been exposed to Holocaust. So there is now really quite a lot of evidence in human for a link between the expression of symptoms in people and what their parents or grandparents have been exposed to. In relation to trauma, there are a few studies linked to Holocaust survivors, which have also started to look at epigenetic factors, even if it's only correlational. I think because epigenetic factors, in this case, DNA methylation measured in blood, it's only a relationship between changes in parents and offspring, in parents exposed to Holocaust, survivors of Holocaust, and their children. But at least it's a good beginning to start to consider these possibilities for this type of pathologies. In rodents, I mean, the evidence is quite substantial as well, and has grown in the past years. I think 10 years ago, I mean, the topic was hardly discussed or hardly considered, and very often just pushed away. Now, I think every single week there is a paper on epigenetic inheritance, and I just listed a few of the initial or important papers showing that endocrine disruptors. I mean, this paper in 2005 from the lab of Michael Skinner was one of the landmark papers, so published 13 years ago, which showed that the injection of endocrine disruptors, I mean, two compounds in particular, vinclozolin and methoxylor, which are used, are pesticides which are used in agriculture, in particular on vineyards, used in Europe for many, many years. now banned. I mean, this particular one, but there are many other endocrine disruptors which are still in use. But that if they are injected during gestation in a rat female for just 6 days, I think the effects, it affects fertility in the pups, and these fertility problems decrease is transmitted across multiple generations. So there have been also studies on postnatal trauma, our work on fetal alcohol, paternal obesity. So overall, there is a number of studies showing that manipulation in inbred mice, some are in outbred rats, but many of these studies are inbred mice, so mice which are genetically identical or almost identical, that this can induce symptoms and that the symptoms can be transmitted across multiple generations. In most cases, it's just the affected animal which is bred, which is crossed with a control partner. In the initial endocrine disruptors, it's 2 affected individuals which are exposed, so which increase the chance that it is transmitted. But many other studies now, I mean, most studies now just use a control as a partner, a breeding partner, so which the symptoms are expressed only by one parent, and yet most of the offspring express the symptoms. So in the concept of transgenerational exposure, so there is intergenerational, so it's from direct parents to the first offspring, which is often the case if the breeding happens when the individual is directly exposed, is being exposed to the, let's say, the high-fat diet or the trauma, so that at the time of exposure, at the time of breeding, of use of the germ cells, the animal is being exposed. But transgenerational means multiple transmission. So if you consider here, this man is exposed, so not only all his cells may be affected, somatic and germ cells. And if his germ cells are affected and keep a memory, it does not have to be all the changes which are induced by exposure, but some changes induced by exposure, if they are maintained in the sperm cell, if let's say he's breeding immediately after exposure, so the mature sperm cells may carry alterations which could be transmitted to the following generation. It doesn't mean that the offspring may express some of the symptoms linked to exposure, but it does not mean that the germ cells in the offspring will be affected as well. So here in this case, it's a bit different because exposure will affect mother, but also the baby, and potentially the future gametes of the baby. So if these cells here, which are being developed, if they are affected, and again, if they can keep some of these alterations, not necessarily all the alterations, but some of them, so there are now 2 generations down which may be affected. So here, let's say we talk about exposure, so breeding or generation of an offspring or fertilization of a germ cell which is being exposed. Now you can imagine that exposure could have happened during childhood. So now the germ cells, they are being used for fertilization, but many months or years after exposure. So whether this could also lead to a transgenerational inheritance. So this is a completely different question mechanistically, because it means that there are marks which have to persist very long, for many months, and in case of spermatogenesis, so go through multiple cycles of spermatogenesis, but still be there to be transmitted to the following generation. So that's really the concept that we are, in my team, we are mostly interested in, really the long term, the most persistent changes which could explain transgenerational inheritance. So just to come back to this principle, but maybe going even further. So let's say this animal, whether young or adult, is exposed or early trauma, nutritional insult, or endocrine disruptors, but it could be other things, alcohol, cigarette smoke, or different, or perhaps a combination even of these insults, and the germ cells could be affected. Here, this is a picture of the seminiferous tubules with spermatogonia. Well, sperm cells, semi-mature sperm cells, because they released, I mean, they are more mature, the maturation continues, but you have the spermatogonia cells, trying to find my, yes, spermatogonia cells, which are here, and the different stages, spermatocytes and spermatids and sperm cells, which are released there, which will travel in the epididymis where they will finish their maturation. So let's say this exposure is long enough to affect all the stages here. And this is oocytes. And we assume that there are changes which happen, which are induced there and which will persist in a way to be present in the F1, in the progeny, in every cell, basically, except if there is correction and except if not everything is transmitted, which is very possible. So, but let's assume the germ cell C will be affected as well. This has the potential to be transmitted to F2, F3, and F4. And here, if we keep an open mind, as Vincent suggested, I think it's absolutely necessary in this field. Every potential mark or factor or mechanism which can keep a memory of exposure or which can be modified. What is a memory of exposure? It's a modification of something, a factor or an ensemble of factors, which can stay either as exactly the same way as it was modified, or maybe slightly changed as well. But the important thing is that there is a signal somewhere which is maintained. So it could be DNA methylation alone or together with other things, histone modifications and non-coding RNA. So here this is a brain cell. So obviously, what is going to be changed in the brain cell cannot explain transmission to the following generation. It's only if it's modified in the germ cell that there is a potential for being transmitted to the following generation. And here, sperm cells. So the chromatin is a bit different in sperm cells. You see here the classical chromatin with nucleosomes. In sperm cells, at least in most mammals, the most histones are replaced by protamines. It depends in the species. I think it's between 1% to 15%. In human, it's up to 15%, which remain. So there are a few nucleosomes here and there, but the rest of the chromatin has this structure of a toroid. So most histones are replaced by protamines, which are these proteins. So they are much smaller, and they allow the compaction of the chromatin. These protamines, it was recently shown, at least in mice, it's part of our work, shown that the protamines, like histones, can be modified. So by phosphorylation, acetylation, methylation, so there is a protamine code which could keep some information, but it's not known whether this code, what's the relevance of this code, but it's known that it exists. So DNA methylation could be involved, protamines, histones, modifications, so modifications of the remaining histones in the remaining nucleosomes, and non-coding RNA. So non-coding RNA, there is a whole population of non-coding RNA, small, long, and many different small, from piRNA to microRNA, tRNA transfers, SNO RNA. So it's a high diversity of these RNAs, which are quite abundant in sperm cells. And long non-coding RNAs, there are poly-A plus, with and without poly-A. There are transposable elements, transcripts. So there is also a very big nuances of different non-coding RNAs. But they could potentially all together or individually in different combinations, depending on the exposition, exposing factor, could be altered, and the alteration could be maintained. And this could also happen in oocytes. Oocytes, I mean, they don't have protamines, so the chromatin is very similar to somatic cells, but there is also DNA methylation, non-coding RNA, and histone modifications. Yes. So to first address the question of first of all, whether conditions in early life, early postnatal life, can influence mental and physical health severely enough to be transmitted across multiple generations. So it's a question which less than 10 years ago was not really clear. So to answer this question, we developed a mouse model of early trauma, and the mouse model ultimately was also, the purpose was to have a solid enough model to then start looking at the mechanism which could explain germline-dependent transmission. So the first challenge was to generate a model which is germline dependent. So in which condition during early life affect the body, mental health, and the physical health. Well, initially we were aiming for changes in behavior, but realized later that it's not just behavior which is altered by trauma, but many other functions, which a posteriori seems a bit normal, because if the whole body is exposed to trauma, there is no reason that only the brain is affected. And indeed, there are many symptoms associated to the behavioral alterations. And if we have a model which is solid enough to transmit these symptoms across multiple generations, and if we can demonstrate that this depends on the germline, so if we can exclude all other factors like maternal care, social transmission, and all this, what are the mechanisms in germ cells which are responsible for this? So the model is based on, it was very, inspired from human literature around a particular disease which initially was a borderline personality disorder, because it's a disease which is almost always associated with traumatic experiences in early life, in particular physical and sexual abuse. So we used this literature to develop a paradigm which was as close as possible to what happens in human. And this paradigm involves unpredictable maternal separation. So the pups are separated from the mother every day, but at different times of the day for 3 hours. And during this separation, the mothers are also traumatized or stressed. So anytime during the 3 hours which happen anytime during the day, so we expose the mother, either we put her in a tube to be restrained like this for, I think, about 10 minutes, or in a glass of cold water. Either one. So there is a lot of unpredictability in this paradigm, which is extremely important, because in mice or in rodents in general, if this is predictable, if separation happens every day at the same time, the mice after 2 or 3 days, they will know, and they anticipate by giving a lot more maternal care before the separation. So ultimately, you end up with more maternal care than what you expect, which is not what we wanted. So that's why we made it unpredictable. And we tested a lot of variations. So with maternal separation, predictable or unpredictable, maternal separation with maternal stress or not, with unpredictable maternal stress. So we had multiple conditions, and we bred these mice afterwards and picked the paradigm which gave the most severe symptoms and those which were transmitted to the following generations. And more or less as expected, it was the most severe manipulation, unpredictable maternal separation combined with unpredictable maternal stress. So the separation, the MSUS, it's the abbreviation for the manipulation, is done from P1 to P14, so it's all postnatal, way after all these reprogramming of DNA methylation and histone PTMs happen during embryonic development. So the epigenome, at least at the level of DNA methylation and most histone PTMs, is kind of set, although it continues to evolve postnatally. But it's also, so it's not only after all these important epigenome establishment period, but it's also a very short, quite short time window. It's just 2 weeks. And breeding happens many weeks, many months after this manipulation. So here we are really focusing on changes which remain, at least those changes, and there are probably many acute changes which are then disappear, but here we are talking about only those which persist until adulthood, which may be transmitted. So we generate a, these pups are subjected to MSUS. We wait for 2 weeks. The last week before weaning, they are with the mother. There is no stress, no nothing, so normal rearing. And we wean them at P21, and then we let them develop until adulthood, 2 to 3 months. Then we test the behavior, physiology, many things. And we breed them. So either the male, here I'm showing the Patry line, but we also did breed the females. So we breed them with wild-type, naive, primiparous females to generate the second generation, and then we repeat again, we test them and so on, and we breed them to, again, naive females, which are not the same as those, they are new females, to generate F3, and we've done F4 and F5, and we are now generating the F6. We also did the experiment in females and down to the second generation, and many of the behaviors that I will show you, or the things that I will show you in males, are also transmitted in females. We just revived this Matry line to dig more into the mechanism. Now, all of our work in the past 15 years have been done on the Patry line. Here, I'm not showing it, but we did IVF, in vitro fertilization, we did artificial insemination, cross-fostering, so all these strategies to exclude maternal care, the influence of maternal care, of social interaction, or anything like this. And indeed, the males here, they are placed with the females during just breeding for just a few days, and then they are removed from the cage, so they are not even with the mother when she's expecting, and they never see the pups and never encounter them. So there is no possibility of any social transmission between the males, which have many phenotypes. I mean, they are depressed and antisocial and so on, I'll show you. But there is no interaction between the father and the pup, so no possibility for this route to be used for transmission. I'll show you just a few behaviors, just as a reflection of what we've done. But in human, it's known that traumatic experiences in early life make people depressed. So in mice, there are different ways to test depression. This is one way. It's the four-swim task, which was developed in the 70s by Mr. Posselt. So you just place a mouse in cold water, in a glass of cold water, and a normal mouse will swim and try to escape. A depressed mouse will float. So you measure the latency to start floating. They usually start floating sooner than control mice. These mice will ultimately start floating. So we measure the latency, but also the time spent floating. So it's a pretty good reflection of behavioral despair. If you inject this mouse with antidepressant, it will start fighting. It's one task. There are other tasks like the tail suspension task, where you hold the mouse by the tail. It's based on the same principle. A normal mouse will fight and try to escape. A depressed mouse will stop moving, be immobile, and you measure the latency, how long it takes them to start being immobile. So what we observed on this task is that the animals have here, it's the time spent floating. They spend more time floating. There is a very clear depressive-like phenotype, which was confirmed on the tail suspension task. The animals are also anhedonic, so they don't enjoy pleasure as much. If you give them the choice between water and glucose, they will not prefer glucose. So they don't really care. They don't mind. So this trait, anhedonia, is very typical of depression in human, but in this case, it's not transmitted. So depression is transmitted to the third generation. I did not show it here, but we could not see it in the F4 nor in the F5. So it seems to be transmitted a certain number of generations, but not after the third generation. Although we don't know. Depression is a type of trait, even in human, which can skip generations. So we will soon test F6 and see whether it has completely disappeared or maybe it may reappear. We don't know. Risk-taking is also another type of behavior which is very often observed in people who have been traumatized during their childhood. And here, so it's one task which can be used to test risk. It's also a task which is used to test anxiety. Our mice are not more anxious, but they take a lot more risk. So you place the mouse in the middle here, and a normal mouse will just stay in the closed walls, closed arms, because they are walled and they are protective. They spend a lot of time exploring these arms, and at some point they will venture on the open arms and dare to look at them. But overall, they spend more time in the closed arms. So there are many parameters which can be measured: time spent in closed arms, time spent in open arms, number of attempts to enter the open arms, risk assessment behaviors like this one. You know, they look down, and all of these parameters are measured. And what we observed on this task is that our animals take a lot more risk. So they enter the open arms much more readily than the control mice, and they spend a lot more time on them, and sometimes they even run to the end and even fall. So a lot of risk taking, and the risk taking is transmitted quite consistently down to the F4 generations through the males. We recently obtained F5 data. It seems that in F5 it's not present like this if we test them without any prior behavior. If we stress them a little bit, we give them an acute stress before putting them on the elevated place maze, then the risk-taking reappear. So it seems to be a trait which is quite consistently expressed across generations. And in F4, it's essentially it's in both females and males. So some behaviors can indeed be transmitted across multiple generations. So we also tested the social behaviors which are affected. The animals are antisocial. Memory is affected. So there are many behaviors which are compromised. Not always negatively. Maybe I'll say this now. It could have been a question, but there are. There is stress resilience which exists in human and also in mice. And what we have observed in our mice is that they are better in very challenging situations. There are tasks like this where it's complicated. They are thirsty. They need to understand a task, press a lever when a light turns on or off, and there is time. There is the rules of the task, so it's difficult. And our mice are better. They are able to find resources or develop a strategy to get the reward, which is complicated to get. They are better than other mice. So they have this ability to be better in very challenging situations. So not all the consequences of this trauma are negative. So as I said earlier, it's not only brain functions which are altered. Metabolism is quite severely altered. So in F2, there is decrease in insulin. If in F1 there is both insulin, in F1 there is also glucose which is altered, but both glucose and insulin are downregulated. And even after an acute stress, normally an acute stress, you see it here, so that's the baseline level of glucose. It increases because there is a stress, and then it comes back to baseline. If we wait long enough, it will come back to baseline here. So even after such mounting of the glucose response, the mounting is normal, but the level of glucose always is lower. So altered glucose and insulin in these animals in F2 blood. But not just glucose and insulin. We did mass spectroscopy in vivo in these animals across generations, and it's a very nice technique because it's non-invasive. You can measure things in your mice, and then you can use them afterwards because it's in vivo, and it also allows to measure many different metabolites from glucose, glutamine, lactate, alanine, and so on. And you see that there are many of these metabolites, including cholecysteine, I think, glutamine, which are and aspartate, which are altered. So an ensemble of metabolic alterations. We recently found that lipids are also altered. There is a quite severe dyslipidemia in these animals, which fits with what is observed normally in a human with depression. So many different symptoms, most of which are transmitted across generations. So what could be the mechanism? And here we've not really just focused on one mechanism, but we have tried to look at them not together, but one after the other, but never ignoring any of them. So regarding DNA methylation, it's work which is still in progress, but initially we focused on a few target genes which were relevant for the phenotypes, and one of them is MECP2. because we knew that MECP2 expression was altered in the brain in exposed animals and their progeny, and also because MECP2 is an important epigenetic regulator. It's a protein which can bind to methylated DNA. It's been linked to Rett syndrome. It is linked to Rett syndrome in human. It's an X-linked gene. So we, by using bisulfite power sequencing, at the time, only on the CPG island of the promoter, because that was more than 10 years ago, and at the time there was much less known about DNA methylation and the importance of other parts of the gene than the promoter. So that's why we focused on the promoter. We are now doing whole genome bisulfite sequencing to have a much better view of DNA methylation across the genome. But what we found at the time was that there was a hypermethylation, small, a few percent, but still on several CPGs. So each bar here represents a CPG. Increased DNA methylation in F1 sperm, so in the sperm of males which were directly exposed to this MSUS manipulation when they were pups, and hypermethylation in exactly the same stretch in the brain of their progeny and in the sperm of their progeny. Here the data are expressed differently. Here it's methylation differential, so control versus MSUS. Here it's the real percent of methylation, and you see that there is too much methylation on some CPGs. And this correlates with a decrease in the level of expression at the mRNA in the brain of the progeny. So a change which is found in the sperm of the exposed male and in the brain and sperm of the offspring. Too much DNA, too much methylation on some CPG in some sperm cells. But there are other genes like CRF receptor 2, so it's a receptor for one of the stress hormone, and it's a receptor which is mostly involved in stress coping. And here it's the opposite. There is not enough DNA methylation. I mean, there is the absence in some CPG in some sperm cells are missing a methylation because it's a population methylation. Here we have tens of thousands of cells. But you see that there is a hypomethylation again in F1 sperm, in the brain of their progeny, and in the sperm of their progeny. In this case, CRF receptor 2 is a bizarre gene because we found at the time, we were very surprised that less methylation correlates with less expression in the brain of the exposed males and their progeny. So, which is a bit counterintuitive because DNA methylation is normally known to be a silencing mechanism. But we now know that it's maybe CRF receptor 2 is one of the exceptions for this. So what we did at some point also was a clonal analysis. So here it's a population, so it's many sperm cells which are measured at the same time, and we wanted to know whether there are, well, a reviewer wanted to know whether there is a difference in individual sperm cells, so we did clonal analysis, and what we observed was that globally there were changes, significant decrease in this stretch, but that each individual sperm cell may have a different profile, a different pattern of DNA methylation. As long as on this stretch there was a few percent decrease, it seemed to be sufficient to be to transmit. At least, we don't know. We looked only at promoter. It could be that there are changes elsewhere. We don't know. But at least there was a representation of different changes in different CPGs. So other genes are not altered, even if they are relevant, and some of them are 5-HT1A receptor. For instance, it's a serotonin receptor. We know that its expression is altered in the hypothalamus of these animals across generations, but it happens that the promoter of the gene is not altered at the level of DNA methylation. Again, does not mean that there is no alteration at all. It could be elsewhere on the locus. We don't know yet. Now, all this is correlative. So using the CRISPR-Cas9 system, we recently assessed. The idea was to mimic these changes using this system, which we use the fusion between the deactivated Cas9 and DNMT3, so to induce DNA methylation where we want, depending on the guide RNAs that we use. And we picked the promoter of MECP2 and the promoter of CRF receptor 2 because we knew that the level of DNA methylation was altered. This is in N2A cells for the moment. But you see that each color here is a different guide RNAs. So we use 3 combination of guide RNAs, and you see that this increased the level of DNA methylation quite substantially in the promoter. And this correlated with a decrease in MECP2 expression. So which is good. CRF receptor 2, remember there was a hypomethylation and downregulation. So now with DNMT3 here we increase methylation, which is obtained on some CPG, and we observe that this increase in DNA methylation correlated with an increase in expression. So which somehow confirms our observation, our correlated observation that an hypomethylation of the promoter was associated with a decrease in the expression. We tried DCAS9 fused with TET, which to obtain a demethylation, because TET is they contribute to eliminate or oxidize the methyl residues, but the construct did not work. We could not recapitulate, we could not reproduce demethylation. We don't know why. It could be due to the sequence. It may not work very well in promoters. We don't know. But we hope to be able to do this one day, maybe in germ cells, to see whether in germ cells after development, after spermatogenesis, for instance, this could be maintained. Indeed, if the idea is that DNA methylation may be altered, I mean specifically depending on the locus, we checked whether DNMTs are deficient or are modified in a way in spermatogonia, which are the cells, the stem cells of the sperm cells. And we collected spermatogonia from postnatal day 7 pups. So after 7 days of trauma, or after 14 days of trauma, it's necessary to work that early. First of all, because our paradigm is between P1 and P14, and we wanted to know what is the consequence, what are the consequences of this trauma at this time of exposure, and also because at this stage, the seminiferous tubules here, they contain essentially spermatogonia. It's the spermatogenesis, or the differentiation of spermatogonia into spermatocytes, start in mice after PND 12-13. So it's convenient because you don't have to use any marker, anything. You just you can sort the cells using a few by fax few markers on spermatogonia, but you don't need to GFP label your spermatogonia, for instance, which you do need to do in adults if you want to collect spermatogonia in adults, because there are many more different cells in testes than just spermatogonia. So, and we observe that DNMTs seem to be altered by trauma in spermatogonia or in testes, which is an indication that DNA methylation may be altered by trauma. So, another potential mechanism may be through non-coding RNA. So we looked at this initially by running RNA sequencing on the sperm of the traumatized males, and we did small RNA sequencing and long RNA sequencing, and we observed that both populations are altered significantly. So each, I mean, we did it not single cell yet. These are bulk RNA sequencing. So on 3 samples here, each sample contains the sperm of 5 males. And here we did the experiment twice. So this is the first experiment, so with controls and MSUS. The second experiment, just because RNA sequencing can sometimes be a bit variable. So, and we had difficulties in confirming, validating some of these data. So we decided to do it a second time. And the data are somewhat consistent, not completely, but just to show you that both small and long RNA are altered, which was expected because there is no reason for one population to be more altered than another. And initially, we focused on validating small RNAs, so microRNAs. And there are a few microRNAs which are consistently altered across the body. So they are overexpressed in F1 sperm for most of them. Many of them are downregulated. It's the same microRNAs, just the level of change, which is different. They are downregulated in serum. We are not the first to observe this, that microRNA which may be increased in brain, for instance, they are often decreased in serum, except this one, the 375, which is wherever we look, it's overexpressed and sometimes hugely. And in the hippocampus also, some of them are overexpressed. So this is in F1 males, so the males which were directly exposed to trauma, but their progeny also had some alterations. you see that in F2 serum, there are 2 of them which are altered, and there are several which are overexpressed in F2 hippocampus, but not in F2 sperm. So despite F2 sperm being able to transfer to the F3. So it suggests that non-coding, at least microRNAs, these microRNAs, they are probably not vectors of transgenerational inheritance. They may carry a signal of trauma, which is spread everywhere, which is transmitted to the following generation, but not to the germ cells of the following generation. So the signal may be transferred to some other factors, or maybe these microRNAs are just not involved in inheritance. We don't know. We have not yet looked at our non-coding RNA in F3, but I think we'll do it at some point. Now, in terms of causal evidence, because this is only correlative. So what we did was, unlike DNA methylation, which is very difficult to recapitulate, today, technically, it's not really possible to induce a little bit of hypomethylation here, a little bit of hypermethylation somewhere else. It's just too complicated. We need specific guide RNAs, and the system nowadays, it's not specific enough. Even if you have guide RNAs, there is always changes in DNA methylation outside of the sequence, which is supposed to be covered by a guide RNA. So technically, it's not possible to reproduce the changes and see the consequences. For RNA, it's a bit different. It's possible to manipulate RNA in an egg. And in our case, since microRNAs were overexpressed, we thought we just could simply inject sperm RNA into an egg, fertilized egg, and see what it does. So that's what we did. We took a control fertilized egg and injected total RNA from traumatized males, and after injection, we transfer the eggs into a pseudo-pregnant females to generate an offspring. Then we breed this offspring to generate the offspring of an offspring. And what we observed was that most of the symptoms were reproduced in these animals here, which arise directly from the sperm RNA-injected eggs, but also their progeny. You see here the depressed phenotype, the risk-taking is reproduced, not only in them, but also in the offspring. So the offspring of the control injected with the RNA. Here is the depressed phenotype. And not only behavior, but also this alteration in insulin and glucose. It's even stronger because remember the insulin was altered only in F2. Here it's directly in these animals, directly injected animals. And glucose it does not even mount the response after stress. It's completely down. So quite severe phenotype reproduced by simply injecting the total RNA from the sperm of these traumatized males. Now, which population of RNA could explain this change? Because there are small RNAs, long RNAs. So we repeated this experiment by partitioning small RNA, so whatever is smaller than 200 nucleotides, and whatever is higher than, longer than 200 nucleotides. And what we observed was that there are a few things which are reproduced. For instance, here on the risk-taking, I mean, there is a risk-taking which is almost significant with a long partition. So whatever on these graphs on the left is it's small RNAs, controls MSUS, long RNAs, control MSUS. So nothing really on the depressive phenotype, nothing on the risk-taking. Here, light dark box, it also tests the risk-taking in a way. It's a trend for the long RNA, but something for the small RNA, but oppositely to what we expect. So overall, behaviorally, it's some things are reproduced, but which don't fit with the normal phenotype. So it means that small RNA alone, long RNA alone can't do anything. It's just they both need to be together to really produce the phenotype. For the glucose decrease, in this case, this decrease is reproduced by the long RNA fraction. The small RNA fraction does not give anything. So our conclusion is that small and long RNA really need to be together to really reproduce the phenotype. So it's a very important finding we find, even if it's not maybe what we would have. No, we did not expect anything, but there are people who have demonstrated that if you inject a few microRNAs, like we did in fertilized oocytes, There is one paper using a combination of 9 microRNAs. They can reproduce one symptom. I'm not sure. In light of our data, I'm not sure if a few microRNAs could really do it. We know that in our model, which is a complex model, I admit, but in our model, it's not just a few microRNAs which are responsible. It's really the mixture between all the microRNAs and other small RNAs, including perhaps tRNA fragments. We don't know, but it's really the combination of both populations. So I have a white slide here to remind me to say that the couple of next slides are unpublished data. So if the camera could just maybe not show them immediately, whenever this is published, I have no problem. But for now, I'll just share them with you. So now one very important question in the field or for the concept is, whatever are the epigenetic changes or the factors which are altered, how are they altered? What are the signals which tell DNA methylation to be more here, less there, and RNA, non-coding RNA to be altered? So because in this case, the whole body is affected, and we know that there are changes which are similar in brain and sperm and blood, that one of the factors which is in common between all these organs is blood itself, because it circulates. So the idea that we developed is perhaps there are factors in blood which are induced by exposure to trauma, and which could send these signals, the same signals or perhaps different signals, to different organs, and that's why you see epigenetic changes in these different organs, which may be the same, different, or some may be in common. So what we thought is that maybe start to answer this question, we would need to know what does the blood of traumatized animals contain. So the blood of traumatized animals during trauma and many months after trauma, when they are bred, when they are adult. So to do this, we did whole proteome and whole metabolome analysis, and I'll show you some of the results a bit later, but we thought, could it be that if indeed the blood contains something, we could reproduce some of the symptoms by just injecting blood? So we prepared animals, so traumatized and controls. We took their blood, we bred them, so to obtain regular offspring and control for comparative purposes. So we took their blood, and what we did was to inject IV the blood, well, the serum, twice a week for 4 weeks, and then wait a couple of weeks, and then breed them, and look at the offspring, and see whether we could reproduce some of the symptoms. And indeed, we can reproduce some of the symptoms, in particular the metabolic symptoms. So there is a decrease in weight. Yes, I did not show this to you before, but in the offspring, up to weaning, there is a decreased weight. And this decrease in weight could almost be reproduced in the animals which were in the offspring of males which were themselves injected with blood. But these offspring had the decrease in glucose, as you can see here. No, that's the regular that's the offspring of the regular control and MSUS. That's the offspring of the males which were injected either control serum or MSUS serum. So you see that the decrease in glucose is reproduced in the offspring of males which were injected with blood. So what in blood could be explaining this effect? The result of our metabolomic screen, so this is a very simple table, but it took us, I think, about 3 years to really get to this, because when you do metabolomic and proteomic, you have tons of data. Some of them, depending on the mass spec that you use, some of these may be true, others, it's difficult. You simply cannot validate metabolomic data like you can do with RNA-seq. So we struggled for a long time with this data, but ultimately we came up with this table where we could show that fatty acids, long fatty acids in particular, polyunsaturated fatty acids, are decreased in the pups which are being traumatized, but then they get increased perhaps as a compensation in the adult. But there is an excess in polyunsaturated fatty acids in these animals, a decrease in bile acid biosynthesis, which is downstream of fatty acids, and an altered, well, a hardly significant alteration in steroidogenesis, which is significant in the offspring. So massive metabolic alterations. So one of the ligands of fatty acids and of derivatives of fatty acids are PPAR receptors, which can bind to LXR, FXR, so it's a complex of receptors. So, which involve also retinoic acid receptors. So we thought that perhaps these receptors are also altered, if indeed fatty acids are altered, whether decrease or increase, but they are altered throughout life, in the adult as well as in the offspring. There must be something about the receptors of these fatty acids. And indeed, when we look at the at PPAR, alpha gamma, there are several of them, and all the other partners of PPAR, as well as some of the target genes, we found that all of them, except maybe this one, are altered. So, and there is a tendency for PPAR to be increased in liver, in sperm, and in white adipose tissue, where PPAR alpha is predominantly expressed in adipose tissue. So it seems that this increase in, or this alteration in fatty acid has altered the signaling pathways. And indeed, if we mimic the PPAR activation using tesaglitazar, it's a PPAR alpha gamma agonist, we can reproduce some of the symptoms in the offspring and in the grant offspring. So the reduced weight can be reproduced, and the injection profile, the injection here follows exactly the injection of the blood, so, which is twice a week for 4 weeks. So reduced weight, glucose is altered in the offspring and in the grant offspring. Also a little less, because it's a grant offspring, but there is still something which is reproduced by these PPAR activation during 1 month. Yes, so that's it for the moment in mice, and we are trying to dissect out a little bit more this pathway. We believe that it's not the only pathway signaling which is in contributing to the alteration in the epigenome, and indeed we still need to find the link between this and epigenetic marks. I will finish with a few slides in human, because I think after all this is nice, but is it relevant to human? So what we did, I mean, there are a number of colleagues who have looked at epigenetic marks in Holocaust people and their offspring. But what we did was to assemble our own cohort, because often, well, first of all, it's difficult to assemble cohorts. And also, there is all the genetic heterogeneity in human, which is always a paradigm or parameter which is complicating analysis. So what we did was to go to one of the countries on the planet which has the highest consanguinity, which is Pakistan. Conveniently, I have a postdoc in my lab who is from Pakistan, from Lahore. He is an MD PhD, psychiatrist. And what he did was 2 years in a row, I mean 2 years in a row, he's now back to there to complete the and extend the cohort. But he went to SOS Children's Village in Lahore and assembled a group, I mean, together with the authorities. It's, to get ethical approval in Pakistan is much easier than here in Switzerland. So he assembled a group of orphans and a group of controlled children in the same school, same neighborhood, many things similar. And these children, they are very similar to our mice because they have lost their father and were separated from their mother. And he did a very long psychometrics in these children and found that they are anxious, depressed, and there are many symptoms, I mean, which is quite common for orphans who have gone through such trauma. And he took blood and saliva and very nicely could reproduce some of our findings in mice in terms of the metabolites. You see in the serum there is an increase in this arachidonic acid metabolism, an alteration in the steroidogenesis as well. And he could also reproduce some of the alterations in microRNAs, in serum and saliva. So, which is very encouraging to us because it says that at least there are some epigenetic factors, if we call microRNA epigenetic factors, but some factors which are consistently altered in mice and in human. we are very far away from analyzing germ cells in human, but I'm trying to work with reproductive biologists in IVF clinic, for instance, to kind of have men consulting for infertility or suboptimal fertility do psychometrics exactly the same way as children here, to try to associate, to see who has been exposed to trauma during the early life. But it's something which is not necessarily easy and will take more time. I had a few slides, but I'll skip them. So I showed you that DNA methylation and RNA, small and long, possibly other factors. I mean, you mentioned chromatin organization, chromatin looping. I think we have to really consider all possibilities here. So I showed that DNA methylation and RNA are altered by trauma exposure in germ cells, adult, and we now have data showing that they are altered already in spermatogonia, in brain, in blood and saliva, and probably we have also data in liver that I did not show, and in some cases across generations, that RNA alterations in sperm are causally linked to the behavioral and metabolic symptoms across generations, but circulating factors can recapitulate some symptoms of trauma, could be involved in the mechanism. We don't know exactly, I mean, yet what it is, but fatty acid signaling seems to be involved, at least in our model, and that some of our data are relevant for human because we can reproduce some of them in children exposed to trauma in conditions where we really selected these children and we can somehow decrease the influence of the genetic background because they are all, we checked this actually, they are all second or third cousins, so we did not do we did not genotype these children, but all of them are from second to third cousin marriage, so the level of consanguinity is quite high. So I'd like to thank the multiple lab members. These are the three ladies who did most of the work. So Isabelle Weiss, at the very beginning, more than 15 years ago, developed the paradigm. Tamara Franklin was the first one to show the transmission of DNA methylation across generations. And Cathy Gap did all the work on RNA. And this is my team at the moment. And we have a number of collaborators. I'd like to thank in particular Mrs. Saba Faisal from the SOS Children Village, who made our life a lot easier than a lot easier that if we would have done this analysis here, and some of the fundings that is supporting our research. And I thank you for your attention. Sorry for going over my time.
So we have time for one or two questions because we are a bit late, but maybe.
Thank you very much. What is the proportion of mice who have the behavior changes at F1, F2, F3? Is it homogeneous, or are there a proportion of affected or non-affected? And my second question is, can you recapitulate the behavior changes with blood experiments or not?
Not quite. There are some behavioral alterations, but they don't necessarily fit with what is induced by the trauma directly. We see alterations in risk-taking, or we see anxiety, for instance, which we don't see in the MSUS model normally. So that's why, blood can recapitulate a few things, metabolic alterations, which seems to be quite, more intuitive than behavior. But we have not really dug too much into this. And, we tried one type of injection, 2 injections per week for 4 weeks. It could be that if we do this more extended or in a different time period, we could see more behavioral changes. And concerning the question of the proportion of animals, yes, most of the animals, if not all the animals, are affected. There is no decrease in the number of animals which are affected. The severity of the symptoms, they are the same. They even increase in the third generation, but then some symptoms, like I showed in F4, depressive symptoms are not present anymore, nor in the F5. So it's possible that some of the symptoms decrease, but those which stay, they stay at least constant, affect the same number of animals. The data I showed you, they are not sorted. It's all the data, and the F4 data I've showed you, you may have seen, no, you did not, I did not show this, but we have an F4 study with up to 125 animals. So you see the blood, Dot plot, not the dot plot, the graph with all the dots. I don't remember the name exactly, but you see all the dots, and they are quite. I mean, there is no spreading. I mean, most of the animals do show the phenotype.
Thank you.
Thank you. I just wanted to make sure I understood. So when you did the several days of blood injections, and how many? It was 12 days of blood injections from the stressed animals. That repeated the epigenetic effects?
We don't know yet. We don't know yet. We have not
So what
yet.
effects did it? It was just in conspecifics that it. So in other words, if you took the stressed animals, took out their blood serum, injected it into other animals, it would recapitulate the effects.
Some of the symptoms are recapitulated, yes, by this by the injection of the blood.
And have you looked at epigenetic?
Not yet. No, not yet.
Okay.
Yes, we will. We have RNA sequencing data in the pipeline of sperm.
Excuse me. Did you look at extracellular microvesicles which could carry RNAs in the blood?
Yeah, we are looking at this at the moment. Yes. Yeah, because the idea, I mean, EVs are would be extremely powerful mode of transport of RNA across the bodies, and it's known. Recently, there were a few studies showing that EVs produced by fat, by adipose tissue, can travel and have effects in the liver. So it could be that there are EVs produced by whatever tissue, perhaps testes, we don't know, or even fat, we don't know, which would affect the brain or other tissues. Yes, it's a very important thing to look at. Yeah, quite challenging though.